We've always thought of the blood-brain barrier as this static, impenetrable brick wall. Yeah, biological fortress just designed to keep toxins out of your brain.
Right.
But it turns out to keep that wall perfectly sealed, your body actually has to constantly dissolve it.
Yeah, it is a massive biological contradiction. I mean, uncovering how that contradiction actually works completely rewrites our understanding of human anatomy.
Okay, let's unpack this. Welcome to the Deep Dive. Our mission today is to explore a truly groundbreaking 2026 paper published in the journal Science by Zhu and colleagues. And we are going to unpack every conceivable detail of this paper today.
Absolutely every mechanism.
Right. So we're looking at something called the luminal surface. If you picture the blood vessels in your brain as the pipes of a massive, bustling city, the luminal surface is the innermost lining of those pipes.
It's the actual biological border. The place where the cells of your blood vessels physically touch the circulating blood.
Exactly. It's an enormous security operation.
Yeah.
Like a VIP club, and this surface is the bouncer. And if you unfolded this surface in a single human, it would cover about 1,000 square meters.
Which is just staggering to think about.
It is.
But for decades, we've had this fundamental problem. We haven't really known what these border guards on the surface are actually doing, or how their protocols change from the day we're born until we reach old age.
Because studying these specific proteins inside a living, breathing animal has been nearly impossible. It comes down to a problem of isolation.
Exactly.
Like if you just take a sample of brain tissue and analyze it, you're getting the entire city block. You're getting the neurons, the structural support cells, the inside of the blood vessel cells, the outside.
You just get everything.
But to understand the security border, you only want to see the proteins facing the blood.
Right. That's the challenge. Previously, to tag these specific surface proteins in living tissue, scientists had to rely on really complex transgenic breeding.
Which takes forever.
Oh, yeah. They had to genetically engineer mice so that their blood vessel walls would naturally express a special marker enzyme. It was incredibly slow, took months or even years of breeding, and you were locked into using only a few species, like mice or fruit flies.
Right, which means we couldn't easily study this in animals closer to humans.
Yeah.
But Zhu and his team solved this with a new tool. I like to call it the biotin paintbrush.
I love that analogy.
Yeah, so instead of breeding new mice, they found a way to chemically paint the inside of the pipes while the blood is literally still flowing.
And the chemistry behind how they did this is just brilliant in its simplicity. So they started with a protein called WGA, or wheat germ agglutinin.
Okay.
And WGA has a very specific natural property. It binds really tightly to certain sugar molecules that are naturally plastered all over the luminal surface of our blood vessels.
So it acts like a chemical anchor. Like sticky tape.
Exactly, sticky tape. And attached to that tape is the paintbrush itself, which is an enzyme called HRP, or horseradish peroxidase.
Got it.
So the researchers inject this tape and brush combination directly into the bloodstream. The tape sticks to the sugar coating on the vessel walls, holding the paintbrush firmly in place right at the border.
And then they send in the paint.
Right. They perfuse a chemical mixture containing biotin XX phenol along with just a tiny amount of hydrogen peroxide.
Okay, so what does the hydrogen peroxide do?
It acts as the catalyst. When the peroxide hits that HRP paintbrush, it triggers a reaction that converts the biotin mixture into phenoxyl radicals.
Radicals.
Yeah. And radicals are highly unstable reactive molecules. Their instinct is to instantly grab onto whatever is closest to them, covalently tagging or painting those nearby surface proteins with biotin.
Okay, let me push back on this for a second, because I'm sure you were wondering this too at home. If you are generating highly reactive, unstable radicals directly inside a blood vessel, why doesn't this act like a microscopic chemical grenade?
It's a fair question.
Right. Like, why doesn't it just burn a hole through the cell membrane and tag all the internal machinery of the cell, too?
It's a completely valid concern. And it all comes down to the lifespan of the radical. These phenoxyl radicals have a half-life measured in nanoseconds.
Oh, wow. So they decay almost instantly.
Yes, they decay so incredibly fast that their radius of effect is microscopic. They literally do not have the time or the stability to drift away from the paintbrush, let alone penetrate the cell membrane.
That is fascinating.
Yeah, they only survive long enough to tag the proteins directly adjacent to them on the luminal surface.
That is an incredibly elegant proximity fuse. And the researchers proved this, right? By running biochemical assays.
They did. They looked for highly abundant internal cell proteins. Things like GAPDH, which helps run the cell's internal metabolism, or actin, which makes up its internal skeleton.
And what did they find?
Absolutely nothing. The internal machinery was completely untouched. Only the proteins facing the blood, like ATP1I1, were painted.
Amazing. And because this method doesn't require any genetic engineering, they didn't just map the mouse brain, did they?
No, they didn't. They successfully used this chemical paintbrush to map the blood vessels in the mouse kidney, the intestines, the muscle. They even mapped the luminal surface of the northern tree shrew.
A northern tree shrew, which is evolutionarily very close to primates.
Exactly.
So we now have a tool that can map the internal plumbing of practically any vertebrate just by injecting it into the bloodstream, which is a massive leap forward.
It really is.
Okay, we've hit our first milestone here, so let's just pause for a quick 5-minute recap before we dive into the findings. Bullet 1, we learned about the WGA-HRP method, the biotin paintbrush, which perfectly and safely tags the inner walls of blood vessels without needing genetic engineering.
Perfect summary.
Thanks. So for you listening, I have a question to think about. If you could instantly paint and map the inside of any organ's blood vessels in your own body, which one would you choose to study first? It's pretty wild to think about.
It is.
Okay, so moving on to section 2. Now that we have the paintbrush, what do they actually find when they turn it on the brain?
To understand that, we have to look at the timeline of a living brain. The researchers used a mass spectrometry technique called 18-plex TMT.
Tandem mass tagging, right?
You got it. Functionally, you can think of this as a highly advanced molecular scale. It allows scientists to weigh, count, and compare every single painted protein across 18 different samples, all at exactly the same time.
Wow.
Yeah. So they compared 3 distinct life stages of mice: 14-day-old neonates, 12-week-old adults, and 80-week-old aged mice.
Okay, so babies, prime adults, and elderly mice.
Yeah.
And the shift they found from the 14-day-old infant to the 12-week-old adult is what they call the maturation transition.
Right.
When you look at the data, it looks exactly like a city transitioning from a chaotic, booming construction site into a mature, highly regulated metropolis.
That's a great way to picture it, because in the developing brain, the luminal surface is absolutely covered in construction equipment.
What kind of equipment?
Well, we see a massive spike in angiogenesis factors, proteins like ENG and CDH5, plus flow sensors like piezo1.
Oh.
These are the specialized proteins required to physically build new vascular roads and branch out the blood vessel network.
Makes sense. The brain is growing.
Exactly. And alongside that, the neonate's borders are packed with nutrient importers, specifically carriers designed to pull amino acids from the blood into the brain. The primary goal of the infant border is rapid expansion and raw material import.
But once that mouse hits 12 weeks, that construction equipment effectively vanishes.
It drops off a cliff.
The roads are built, the brain is fully grown, and the metropolis completely shifts its priorities. The nutrient importers dial way back. And in their place, we see a massive upregulation of what are essentially cellular trash compactors.
Yes, efflux transporters, specifically proteins called ABCB1 and ABCG2.
Yeah, what do?
Their specific job is to grab metabolic brain waste, foreign chemicals, xenobiotics, toxins that have built up in the brain tissue, and physically pump them out into the bloodstream so they can be cleared by the liver and kidneys.
So right now, as you're listening to this, your own brain's plumbing has completely abandoned the rapid import strategy you used as a baby.
Completely.
Your borders are now almost entirely focused on waste management and security.
Yep.
Which naturally makes me wonder, if the adult brain relied so heavily on these active export pumps, what happens when those pumps eventually break down from overuse?
Well, that takes us directly into the data from the 80-week-old aged mice. And the findings here fundamentally challenge how we view vascular aging.
How so?
The researchers identified a 3-pronged transformation. So the first prong, as you predicted, is a drop in metabolism.
Okay.
Those crucial waste removal pumps, ABCB1 and ABCG2, they decrease in number. Plus, vitamin transporters like SLC5A6 and SLC7A1 also drop. The plumbing basically gets sluggish.
Right. The city's waste management starts failing, and the second prong,
An alarming spike in inflammation.
the door guards get aggressive.
Exactly. Proteins that normally tell the immune system to stay calm, like CD200, they completely drop off. Meanwhile, pro-inflammatory signals spike.
Like which ones?
Things like VCM1, MCM, and C4b. These are adhesive proteins that physically catch white blood cells circulating in the blood and pull them aggressively into the brain tissue.
Wow. Okay, and the third prong?
The third prong is mechanical. The blood vessels become physically rigid.
Okay, here's where it gets really interesting to me. I always assumed that vascular aging was a passive process. Just the physiological equivalent of an old rubber band losing its elasticity over time.
Right, that's what a lot of people think.
But the protein map shows this is a highly active structural lockdown.
It really is. If we connect this to the bigger picture, the aged vessels actively overproduce extracellular matrix proteins like laminins, and those act like biological concrete, stiffening the vessel walls.
Like they're intentionally paving over things.
Yes, but perhaps more importantly, the vessels lose their mechanical sensors. Take piezo-1, which we mentioned earlier, or LRRC8C, which senses osmolarity.
Piezo-1 is the one that feels blood flow, right?
Yeah, right. It physically feels the force of blood rushing through the vessel and tells the surrounding tissue to dilate or constrict.
It's a microscopic flow sensor.
Exactly. And in the aged brain, these sensors practically disappear. The vessels don't just become stiff, they literally lose the ability to feel and adapt to changes in blood pressure.
Oh, wow. And at the same time, proteins related to high blood pressure like ACE and CACLA1H actually rise.
They do.
So aging isn't just passive decay. It is a highly active, measurable shift toward mechanical stiffness, high pressure, and severe inflammation.
That's exactly what the data shows.
Okay, time for another quick 5-minute recap. Bullet 2 on our journey. We've learned that the developing brain focuses on building roads and importing nutrients, while the adult brain focuses on exporting waste. But aging blood vessels actively become stiff, inflamed, and lose their adaptability. So asking you again, the listener, have you ever thought about how your own brain's plumbing completely shifted its priorities from when you were a baby to right now? And considering these specific changes, how might this alter our approach to treating age-related diseases? Like, should we be targeting the vessel walls before the brain tissue itself?
It's a profound question, because mapping the proteins is one thing, but how do we know which of these proteins are actually holding the border wall together?
Right. Observation versus manipulation.
Exactly. To figure that out, the researchers had to manipulate the system. They utilized a highly specific viral vector. It's an adeno-associated virus called AAVX1.1.
AAVX1.1. And the beauty of this specific virus is that it has a homing beacon for endothelial cells, right? The cells that make up the blood vessel lining.
Yes, it has a strict tropism for them.
So the researchers could inject this virus into the bloodstream, and it would deliver CRISPR gene editing tools directly into the luminal surface of living mice, allowing them to just knock out specific proteins on demand.
Which is incredible tech on its own. So they started by targeting that transporter protein we mentioned earlier, SLC7A1. And this uncovered what we can call the neonatal paradox.
Okay, walk us through this.
Based on their map, SLC7A1 is highly expressed in babies, but practically non-existent in adults. So they used the CRISPR virus to delete SLC7A1 in neonatal mice.
And to test if the barrier was still intact, they injected a blue tracker dye, Evans blue dye, into the bloodstream. Normally, this dye stays strictly inside the pipes.
Right.
But when they knocked out this one specific importer protein in the infants, the dye flooded the entire brain. The barrier completely and catastrophically collapsed.
Just massive leakage.
Right.
But when they performed the exact same knockout experiment on adult mice, the barrier remained perfectly sealed. The loss of SLC7A1 has zero effect on the adult brain.
So, why does removing an importer completely break a barrier in a baby, but do absolutely nothing to an adult?
To understand that paradox, we have to look at the specific cargo that SLC7A1 carries. Its job is to pull an amino acid called arginine from the blood into the cell.
Okay, arginine.
And arginine is the direct chemical fuel for an enzyme called nitric oxide synthase, or NOS3. And NOS3 uses that arginine to manufacture nitric oxide.
Oh, I see. I think of this as the baby's breath mechanism.
Oh, I like that.
Yeah, the infant's blood-brain barrier literally requires the continuous breath of nitric oxide to stay sealed. Without the SLC7A1 intake valve, there is no arginine fuel. And without the fuel, there's no nitric oxide, and the border wall instantly crumbles.
And they proved this. They used their virus to knock out the NOS3 enzyme directly. The result was identical: massive brain leakage in infants and no effect in adults.
Wow.
This proves that nitric oxide isn't just, a byproduct of metabolism. It is an active, vital structural requirement for the neonatal brain.
Which is a brilliant piece of biology. But it leaves us with this huge evolutionary mystery. Why does the brain switch its fundamental security protocols between infancy and adulthood? Like, what is it about a mature, fully grown brain that makes nitric oxide completely obsolete for border defense?
It's a great question, and it forces us to look for the adult equivalent. If nitric oxide is the sealant for babies, what seals the adult barrier?
Right.
So the researchers ran a genetic screen on the proteins that spiked during that 12-week adult phase, and they zeroed in on an enzyme called HYL2.
HYL2. Okay, time for another quick recap before we explain this next huge concept. Bullet 3. The neonatal brain relies on a unique arginine to nitric oxide pathway via SLC7A1 and NOS3 to keep its barrier sealed, a pathway adults just don't need.
Nicely summarized.
So my question for you listening, why do you think the brain switches its fundamental security mechanisms between infancy and adulthood? It's crazy to think we just discard the baby system entirely.
It is crazy, but the adult system they replace it with is even wilder, because this brings us to the core paradox of the entire paper. HYL2 is a hyaluronidase.
Okay, what does that mean?
Its entire biological purpose is to degrade and chew up hyaluronan. Hyaluronan is this thick, gooey, sugar-based matrix that naturally builds up around cells. It's essentially cellular goop.
Okay, wait. Let's consider how hyaluronidase is used in modern medicine right now. Because FDA-approved versions of this exact enzyme, like Hylenex, are used in clinics to increase tissue permeability.
Exactly.
Doctors inject it alongside other medications to dissolve the structural goop, creating physical space so the drugs can penetrate deeper into the tissue.
That is the standard medical logic. The enzyme dissolves the barrier.
Right. Therefore, if you use the CRISPR virus to remove the HYL2 enzyme from the blood vessels, the goop should rapidly build up and the barriers should become thicker, tighter, and more impenetrable than ever.
That's what you would think. But biology rarely follows our linear logic. When the researchers deleted HYL2, the hyaluronan goop massively accumulated on the luminal surface, just as expected.
Okay.
But instead of forming a thicker, impenetrable seal, that specific buildup triggered the blood vessel cells to panic. They rapidly formed fenestrations.
Fenestrations.
Literal, gaping, physical holes straight through the cells. The barrier completely broke down across the entire lifespan of the mouse.
So wait. The action of actively clearing away the structural matrix is what actually keeps the barrier sealed.
Yes.
That is mind-bending.
It is. And to figure out the physical mechanics of why this happens, the researchers utilized AlphaFold.
The AI system that predicts 3D protein structures.
Right. AlphaFold revealed that the HYL2 enzyme essentially has two distinct parts. It has an EGF-like domain, which acts like a structural tail, and it has a catalytic cavity, which is the actual engine room where the chemical chewing of the goop takes place.
So they played genetic Lego to see which part was actually responsible for maintaining the border wall?
Exactly. First, they completely amputated the EGF-like tail and put the modified enzyme back into the mice.
Yeah.
The barrier stayed perfectly sealed. The tail was completely irrelevant.
So then they went straight into the catalytic engine room.
Yep. But they didn't remove the engine. They just made a single microscopic modification. They swapped out exactly one amino acid, mutating a proton donor glutamic acid at position 135. It's called the E135Q mutation.
So one tiny swap.
One tiny swap. It left the overall shape of the enzyme perfectly intact, but it completely disabled its ability to chemically chew the hyaluronan goop.
And the moment they introduced that single disable engine room mutation into the mice, the barrier completely collapsed. Massive leakage.
Yep. That microscopic swap proved unequivocally that the physical enzymatic act of degrading the hyaluronan matrix is the literal mechanism that keeps the blood-brain barrier intact in adults.
That is, wow. It makes HYL2 the paradoxical plumber. It has to relentlessly chew up and clear away this thick, gooey matrix just to keep the pipes sealed tight. If it stops chewing for even a moment, the accumulation tears the pipes open.
Exactly.
This completely reframes how we think about homeostasis. We imagine our body's defense systems as static shields, like a vault door you lock and walk away from. But they aren't static at all. They're more like a treadmill.
Yeah.
The body has to constantly exert energy and break things down just to stay in the exact same place.
And the implications of that treadmill concept are just monumental for medicine.
All right, final recap here. Bullet 4. HYL2's paradoxical role. Actively clearing away structural matrix is what keeps the adult barrier sealed. So think about this. If actively clearing away structure is what maintains a barrier, what does that tell us about the dynamic, constantly working nature of our body's defense systems?
It changes everything. I mean, the blood-brain barrier has historically been the single greatest hurdle in treating neurodegenerative diseases like Alzheimer's or aggressive brain cancers like glioblastoma.
Because we can't get the drugs in.
Right. We have incredible life-saving drugs, but we simply cannot get them past the border guards.
So zooming out here, We've gone from inventing the biotin paintbrush with WGA and HRP to mapping the aging of our brain's plumbing, to uncovering these crazy rules of the bouncers, nitric oxide for neonates, and HYL2 for adults. So what does this all actually mean for the future of those treatments?
By mapping the luminal surface like this, we now have the molecular instruction manual for the border wall. We know exactly which levers to pull.
Oh, I see.
Yeah, instead of using blunt force to bypass the barrier, we could potentially design transient drugs that briefly inhibit HYL2. This would cause the goop to build up and naturally open those fenestration holes for just a few hours.
Wow. And then we deliver the chemotherapy or the Alzheimer's medication through the open holes.
Exactly. And then let HYL2 resume its chewing to seamlessly seal the fortress back up.
That is incredible. We were moving from seeing the brain's plumbing as an impenetrable obstacle to viewing it as an actively managed programmable system. We've learned today that the ultimate brick wall in your brain relies on completely different, highly active chemical paradoxes at different stages of your life, from the vital breath of nitric oxide in infancy to the paradoxical plumber of HYL2 in adulthood.
It really is a masterpiece of biology.
It is. Which leaves me with a final lingering thought for you to mull over. If the most secure, locked-down fortress in your biology, a system we always assumed was a static brick wall, is actually a shape-shifting system that totally rewrites its own rules as you age, what other supposedly static systems in your body are completely shape-shifting under the radar right now?
A truly fascinating perspective, and it highlights exactly why tools that allow us to map biology in real time are going to define the next era of medicine.
Absolutely. Well, thank you for joining us on this deep dive. Keep questioning the brick walls, keep exploring the mechanics of how things actually work, and we'll see you next time.